Welcome back to Synergistic Science. I'm Dr. Julianna Goelzer, and today we're going to be discussing the regulation of chromosome structure. This content comes from Chapter 5 of Essential Cell Biology by Alberts. I will add the link to it in the show notes. But without further ado, we're going to get into how do we actually modify or change chromosome structure. Previously, we've discussed DNA structure. You can go see that video as well as one on chromatin structure and how we actually get mitotic chromosomes as well as interphase ones and some of the histones and nucleosomes that help regulate or keep it from getting supercoiled. So as before mentioned, we can change the structure of the chromatin. Typically, we think of chromosomes in their mitotic sort of structure as kind of little X's all lined up in a karyotype, but that's not the norm. The majority of the time, the chromatin is going to be more free in its interphase sort of state, kind of stuffed into the nucleus. But don't let that fool you. It is still very organized within that state. And some of the things that help control or organize the chromatin are nucleosomes and histones, on which we can wrap the chromatin around. These are proteins that we've seen before in chapter 5. You can kind of see one of the crystal structures over here. And we can modify how tightly the DNA is wound around these proteins based on different chemical groups. And so we have chromatin remodeling complexes. These are enzymes that use ATP energy to change the arrangement of nucleosomes in the eukaryotic chromosome. And so these complexes are able to come in and kind of shift the kind of yellow here nucleosomes to open up a piece of DNA or shift them over to close it up so it's not as accessible for the genes to get expressed. So you can see the ATP remodeling, chromatin remodeling complex here in green, and the ATP is providing the energy. It's getting hydrolyzed, and then it helps kind of shift over the little blue gene where it is located on the nucleosome. Histone modifying enzymes are enzymes that will catalyze the covalent addition of small molecules or some small chemical groups to the side chain of the histone. So typically these groups are methyl or acetyl groups. They get added to very specific amino acids. There's very common locations on the histones. They're usually notated as like H3K 29 or whatever it is. So it'd be like histone 3 at the lysine and what residue with the number. Okay. And so adding these molecules to it can change the charge. And since we know that DNA is negatively charged, if we can make it more negative on the protein, then negative to negative, they're going to be repelled, opening up the DNA. Or if it's positively charged, it's kind of like a magnet. They're going to be attracted to each other and it's going to be bound more tightly. So now there is more to it, but the idea being if we change the charges and we change the slight structures in the protein, we're going to modify how accessible the DNA is. So a little bit more on those histone modifications. You can see the DNA here in red getting wrapped around the yellow nucleosome, which is made up of 8 different histones. We have them notated as H3, H2B, H2A, H4, and there are duplicates of each of them kind of stacked on top of each other. Okay, and they're showing these kind of little strings hanging off. These are the ends of each of those histone proteins. They're known as tails. And so as you get out to the ends here, this is the part that could actually be modified. And kind of what I alluded to before, you can see the residues are numbered and there's distinct amino acids there that can be modified. And so like the P would be adding a phosphate to a serine, the S group here, or you could add a methyl or an acetyl group to a lysine with the K here. And so often you'll hear things like, okay, we're on histone 3, so it'd be H3 K14 acetylation. And so that kind of gives you the address of sorts on the protein as to where that modification could occur. Now, there's a lot of research going into cracking effectively the histone code. So, like, what happens when we get a certain combination of these different chemical modifications? Okay, so here's some of them that have been worked out. Some of them are more nailed down than others. So, you have, like, okay, if I get a acetyl group here at 14, then I'm probably going to get more transcription. Okay, now, just for, I don't know, giggles here, you can see how many question marks there are, which, I mean, if you're looking for something to do scientifically, you can see that there is still more to be done. Okay, so we do have a rough idea of what's going on, but it's not perfectly nailed down, and it's not really a formula, I guess. Okay, so typically, though, if you see an acetylation of lysine, this will reduce the affinity for the nucleosomes or help the chromatin pull away from them. It loosens the chromatin structure and therefore makes it more accessible for gene expression or any of the um, transcription factors to get in and bind and promote the production of RNA. Okay, so a little bit more if we zoom in on one example here. Again, this is the H3 tail modification we were looking at before. If you get a trimethylation or three methyl groups added to this lysine here at residue 9, it forms heterochromatin, which is closing up of the DNA, which makes the genes more silent or less transcription to occur. On the flip side, if you get a acetyl group at that same residue and a trimethyl group at residue 4, this promotes gene expression or opens up the DNA more so you can get more RNA expressed. Right, so that can happen on a small scale, gene by gene, moving particular nucleosomes. We also see it on a larger scale. So an interphase chromosome contains both highly condensed and more extended forms of chromatin. Okay, so if you envision, I don't know, let's say this is chromosome 3, we can look at it from the beginning to the end. Each end, since it's linear, has a telomere on either end. This would be an example of heterochromatin where it's very tightly compacted, probably going to be gene poor. So we're not going to have many genes there. We don't really care that they're closed up. Then you're probably going to have some segments of euchromatin here in the blue and green. There may be some short segments of heterochromatin. And then typically the centromere in the middle where you'd be actually kind of lining them up during mitosis and pulling them apart is typically going to be heterochromatin and gene poor as well. So heterochromatin is the highly condensed regions of an interphase chromosome. Typically, it's not going to have many genes in it, and it's typically not going to be making a lot of RNA. Euchromatin is the opposite. It is going to be more in gene-rich areas. It's less compact, allowing for more transcription factors, more polymerases to get involved to actually make RNA products. Heterochromatin specific histone modifications will allow heterochromatin to form and spread. So yes, we can modify nucleosomes on a case-by-case basis, but there is evidence that if we have a region that is heterochromatin, it will begin to spread kind of like a domino effect to some degree to the histones that are nearby. And so if we look at this image, you can see heterochromatin specific tails are on several nucleosomes, all kind of stacked up, and they're right next to the euchromatin. And then the histone modifications will attract more proteins to modify the histones that are next to them. So effectively pushing another domino and another domino. And so they modify the ones next to them and spread all the way until they come to a barrier DNA sequence that tells it you can proceed no further. One of the places that you might see a lot of heterochromatin outside of those telomeres is in bar bodies. And so if you were to look at a female human cell, this is also true for other mammals as well. you would expect to find two X chromosomes as opposed to a male cell that would have an X and a Y. Okay. Now, if you have two X's, that means you're going to have a higher gene dosage than a male of the same species would have, which could be problematic. You don't want double of a gene that you're not supposed to have double of. Okay. So, how does the cell get around that? It actually inactivates one of the X chromosomes. Okay, so it picks randomly in every cell and it won't be the same throughout the whole organism. Some will pick one, some will pick the other, and one X, so this in this case, they're labeling it as like from paternal and maternal. So in maybe one cell, it goes, oh, I'm going to silence the red X chromosome I got from dad, and I'm going to express the green one I got from mom. Or maybe in a different cell, we're going to express dads and not moms. And so effectively what you end up with is kind of a mosaic of some cells in the female have X expressed from dad, some from mom. And actually, this is what you see in calico cats. So calico cats are female and they kind of have that like blotchy sort of pattern to them, which is pretty similar to what you would expect with the X chromosome gene expression.